Hello everyone, my name is Gabriela Merino and I will present you our work called Predicting Protein Functions with Deep Learning and Multi-Source Data. I am a postdoctoral researcher from CONICET, Argentina, and this work was done in the context of a Cabana secondment that I did the last months at EBI. As you know, predicting protein functions is a challenging problem in bioinformatics. For solving it, there are a number of public repositories having different levels of protein data that can be used. Our proposal is to use a deep learning model for integrating this data. We selected deep learning because it will allow us not only to minimize the data preprocessing required, but also to model the complex relationships that exist between proteins. For this work, we extracted protein data from 4 different sources. We used editing distance between protein sequences, correlation of expression data, and co-occurrence of protein annotations in gene ontology and in reactome pathways. With this data, for each Go subontology, we obtained 4 association matrices relating all the target proteins with those that have annotation in the particular subontology. For obtaining the protein annotations, we have taken into account the gene ontology annotations with manual curation of each protein in each subontology. Then we represented those annotations by means of one-hot encoded vectors in which each element is a term for a particular subontology. The model that we propose is a fully connected deep neural network with 7 hidden layers. Each layer has a dense layer followed by a batch normalization and dropout. Several models were trained by considering as input different combinations of the information from the association matrices presented before. The model output is a probability vector in which each element represents the chance that the target protein is annotated with a particular Go term. In order to model the hierarchy of the ontology, we decided to split the model output for being learned into 4 consecutive layers. Thus, those layers represent hierarchy levels at the ontology up to the output layer that corresponds to the most specific terms. For evaluating our proposal, we used proteins from yeast. For 5,799 proteins, we obtained their sequences from UniProt-KB Swissprot, and expression data at the gene level from a microarray experiment involving 72 samples downloaded from Gene Expression Omnibus. We also obtained the pathway knowledge from those proteins annotated in reactome and the Go annotation for the 3 subontologies: biological process, cellular components, and molecular functions. The experimental setup we used for developing and evaluating our model was based in a grid search step for optimizing the hyperparameters. We used a 3-fold cross-validation, partitioning the dataset into 70% for training, 10% for validation, and 20% for testing. As you can see, this is the list of values that we evaluated for each hyperparameter, and in bold you can appreciate the selected ones. We also consider early stopping for defining the optimum number of epochs for training our models, and we used performance measures commonly used in the CAFA challenges for model evaluation. We summarize our results in this bar plot. Each bar represents the average values of the CAFA performance measures reached by the different models that we have trained and in each subontology. Analyzing our results, we found that the simplest model using only protein sequence data was the worst for the 3 subontologies. Particularly, the F measure reached for molecular functions and for biological processes was lower than 0.4 and closer to 0.6 for cellular components. We also found that when sequence data was integrated with information of annotations co-occurrence in the other ontologies, the performance scores were improved mainly for molecular functions and biological processes predictions. In addition, when reactome was also considered as input, the F measure was improved for biological process and cellular components prediction. Particularly, those improvements were due because of an increment in the recall for biological process and in the precision for cellular components. We also observed that when our models received information from transcriptomics data, they increased the F measure for the prediction of both molecular functions and cellular components terms. Finally, the highest F measure that we found was 0.65, and it was reached for predicting cellular component terms by the model that used all the association matrices as input data. Although we need a deeper evaluation of our model, considering more experiments and even involving different organisms, with our results, we can conclude that protein knowledge represented by association matrices provides useful information for function prediction. We found that as more input data were integrated by our models, better F measures were reached. So deep learning models integrating multi-source data are a promising tool for protein function prediction. Thank you to all of you for your attention.